So going back to your presentation in New Zealand, you discussed, so we're going to move on to the subject of the mysterious something in the blood that seems to be affecting the would I be right in characterizing it as it seems to be affecting the functioning of the mitochondria in some way?
Yeah, I think that would be that comment.
So you had a graph showing the results of some testing that you did on the mitochondrial function when the cells, some muscle cells, I believe it was, were in plasma from ME patients. And I'll add that to the final video so people can see what I'm talking about.
Okay.
There's a few questions about that graph. So first, the readings kind of at time point 0 seem to start for the rest of the graph very high. So the y-axis is intracellular oxygen.
Yeah.
So is that initial dive down from when, is that the difference between when the samples are taken and when they're being basically tested, monitored, however it might be? Why is there that initial dive, which seems to affect both controls and patients?
Okay, so I'll take you back a bit in how we do the test.
Sure. Yeah.
So if you want to look this up, any of the other forums, so it's a probe made by Agilent. It's called Mito Express Intra, and there's lots of information on the website. We published a review around the test about a year ago, so we can take you. So the way the tests work, or it's a protocol really. So we have a oxygen sensing probe. So it reacts to oxygen. It doesn't react in terms of chemical reaction. It's quenched by oxygen. So when oxygen levels are high, the signal is quite low. So the low signal. And then when oxygen is lower, then you get an increase in fluorescence. And so this probe is on nanoparticles. So these nanoparticles are added to control cells. So in this case, muscle, human muscle, primary muscle. We're lucky we've got some primary human muscle cultures that are quite unique. We add the probe to the cells, they take it up, and it goes in the cell. And then we wash away any non-taken up probe. So what you've got now is you've got a readout of what the cell is doing in terms of oxygen. So what you can see on the graph is that we set these up in air. So at atmospheric oxygen, at 20% oxygen. So the dip down is when they go into the machine, which is set at 8% oxygen. Okay. So that initial drop is just a sort of an artifact of moving from 20 to 8, and then it drops down to the machine. And we see that consistently. And we could put our machine in an 8% box, but that would cost too much money. And we can, with other things we do set things up under physical, but we like to work at 8% oxygen because we think that's really important. Most people don't do that. Most people will work at 20% oxygen. You see the same type of effect at 20, but we've got data coming out of my lab that suggests that physiological oxygen is between 5 and 8. And when you work at 20, you get a lot more radical production. You get different data, not as big as glucose, but there's something we need to think about. So we try and run all of our experiments in other areas. So we will culture the cells under normal tissue oxygen. But for these cells, because we've got to set them up outside, they go in. So that sort of tells you about the initial part.
Okay. And
do you want me, Andy, to talk you through the trace? Would that be, because you might have some of these questions coming up. I can talk you through what the trace shows.
Yeah.
Yeah, and then if there's anything I haven't covered, then you can maybe pitch that in. So what the graph shows, there's a dotted line on the graph which shows what 8% would look like.
Yeah, sure.
So one of the things we noticed, which was quite surprising years ago, was that when you put a probe in a cell, if that cell can use mitochondria, it is actually sets itself at a lower oxygen concentration in the air. So we just assume, and people assume historically, that if you want to work under a certain O2 concentration, you just put the cells in an incubator and leave them there, and they'll be at that oxygen concentration. what we've shown for our work on hypoxia is that isn't true. So cells will be much lower than you think. So if you're trying to do a study at 5% oxygen, some cells will be down at 1. So you're not looking at 5% oxygen. So that's important for some cancer work. So on the graph, you can see the dotted line, which is where the cells would be if they weren't doing anything. So if the mitochondria weren't working at all, they would be along that dotted line. So if we poison them with antimyosin A, they'll be on that dotted line. So the cells would sit some way below that. So muscle cells at this density of cells are about 6% O2. So they're working quite well. And what's nice is they work well over 18 hours. So this is a nice real-time assay. What we noticed when we added some of the patients, and you can see that for one of them on the graph, was a big drop. So there's a big drop in intracellular O2, which means that the mitochondria are suddenly working really hard. So the question is, and if you look at the graph, that was sustained for the whole 18 hours. We measure cell death in this model. There's no dying. There's something that's changed by adding the plasma. It's 8 out of 32 patients on the muscle cells. So we've gone on and looked at this in other models. We've looked at other samples. So we've looked at a Spanish cohort from ELISA. So these were EDTA samples like these ones. And the muscle cells are tricky, and poor Eloy was trying to grow them. He came over to work with us on an Erasmus exchange, struggled. So we said, right, let's lose some cancer cells. They're good mitochondrial function. Let's see what you see. And essentially, now we're seeing the controls and the patients do something very similar. So controls on a cancer line can change mitochondria quite dramatically. There is a subtle difference with the cancer cells. It's towards the end, but it's not as clear cut as the muscle. We've also looked at the biobank samples, which are in heparin. The controls and patients are identical. There's no difference. So at the moment, we're not sure what to make of this. So a lot of these samples were brought in for other things. So we've done metabolomics on the samples. They've been defrosted, frozen, defrosted, and frozen. So we wonder whether the differences might be the processing again.
Yeah.
So we hope to start a new project with the ME Association to look at a brand new cohort from Poland. It's never been touched by humans. It's been frozen. We have EDTA, we have heparin, we have serum to really test it to death, and try there's other rodent lines we can use. Focus on muscle, because one of the things I've not mentioned in this presentation is we did a little bit of work with a microscope with a labeled mitochondria, and they move very fast with the patient plasma. So I think there's something in the blood, but it's also potentially in controls as well. Are there subtle differences? So we plan to try and tease this out over the next 12 months when we start the new project. This does fit a bit with the Ron Davis nanoneedle. They've got something in their blood, and something not in their paper is that when they switch the plasma around, so they take the control plasma and give it to the ME-CFS cells, they don't see such a big effect, no effect. And then, so it seems to be the plasma is where the action is. And it seems to be the plasma where there might be a factor that's modulating energetics. But we need to work out why the cancer cells are different. Cancer cells could have something on the surface that's quite different from primary muscle cells. And if we can work out what it is, it might give us some clues. So it's trying to move on from this, really.
So for those confused by a couple of names there, you mentioned EDTA and heparin, which, as I understand, they're both storage solutions. Is that correct for samples?
For blood, yeah. So when you take a blood sample, if you're going to have a clinical test, people will be interested in the serum. So basically, you clot the blood. But that can change lots of factors in the blood.
Yeah.
If you want to get plasma, which essentially doesn't involve clotting, you put the blood in either an EDTA tube, which basically chelates all the calcium and stops platelets clotting, and then you can process a sample without clotting. The heparin also impacts on protein clotting, but more directly at the platelet level. So there are two different ways of preparing plasma, and from our metabolomics work, we think they might do different things. So EDTA could change cell function in the tube. We looked at EDTA in our tests. So we took EDTA from the tubes and added it to the, it does nothing. So it's not the EDTA that's doing anything here, it's something in the tube. So there are different ways of processing blood, which potentially, when you're starting to do more sophisticated biological analysis, could impact on the data. You know, most blood tests at the GP, they just test things and then it doesn't really matter what tube. Well, they have special tubes, but it's probably not a big deal because you're measuring, you know, the amount of X in a sample. So yeah, so that's what the tubes
are. Okay, brilliant. Another question is, so you mentioned the Stanford nanoneedle experiment, which they achieve through adding a salt solution to the cells. Is that something that you could look at using your methods, or is that?
Sure, no, very easy. So when we get this new project going, so we could add a salt stress to the muscle cells, you know, the controlled muscle cells. So basically the Stanford approach, we don't quite know what drives the conductance difference, but essentially if you add too much salt to cells, they will try and get rid of it. And they do that using an ATP process. So it's sort of linked to the mitochondria potentially. So yeah, so we can easily do a salt stress and see what the plasma does to our oxygen consumption data. But we've got a direct readout of the mitochondria, whereas the conductance test, you're not quite sure what's driving that. But obviously we don't have the equipment to do the salt test, but the sodium chloride challenge. What's interesting, if you look at the graph again, if we inhibit glycolysis with inhibitors, we see there's something very similar to that trait. So it dips down massively. So if you block glucose metabolism at the level of glycolysis, so prevent it going into the mitochondria, either with a lactate inhibitor or deoxyglucose, we see something very similar. So that's quite interesting to me because we wonder whether that change could be blocking glucose utilization, which fits with some ideas around ME/CFS.
Yeah, I certainly think that there have been other results that suggest glycolysis is impaired in patients, which from what you say would seem to fit with what you found there potentially.
Yeah, essentially it's just knowing what's going on. You know, if we can what's the factor, you know, we can show the same data with the inhibitor study, but it's knowing what's going on here. So we know that effect is heat sensitive. So if we heat the sample to 55, we lose that effect. So there's something potentially biological. But when I talk to the guys on Metabolon, there are metabolites that are also heat sensitive. So you're not quite clear. Could be a microRNA. Many things can be heat sensitive. So it's interesting, very interesting.
Okay. Another question from the forum members, and one which we'd all have to get a definitive answer on, is: does the test about on the oxygen concentration, does that have potential for use as a diagnostic test? And then my addition to that would be, is it likely to be able to be used as a diagnostic test?
I think we need to test it in other illnesses. That would be my take on this. We need to test it in other conditions that may be similar, like MS, Parkinson's, lymphoma. There are lots of conditions with fatigue. So if it turns out to be specific, but the fact we can see an effect in controls does make me wonder whether it is going to be, but there could be blood cells, you know, muscle cells might be the cells to use. So I think we need to look at more different patient disease cohorts and other illnesses. I think that's really important. Most of the research in ME/CFS has compared controls and patients, which is great, but we need to move, if you're going to have a test, it needs to be specific. And it needs to be robust over time, you know, like the tests with the ATP or the PBMC oxygen consumption test. It needs to be, you know, if I take your blood today, it needs to be the same in a few weeks' time if nothing's changed. So I think we're quite away from the test, and there may be better tests. You know, doing tests on live cells is a challenge. If we can find a test that maybe links to a metabolite, that would be my choice, that's robust. When you start to do things like this, you know, what we've seen in our data, just using different cell lines, can show differences. So I think a test going forwards would need to be something that's usable. Anyone can use, you know, because to make a PBMC prep, you're not going to be able to do that in every lab and every country. So it needs to be something probably more around the metabolomics maybe, but we need to be careful with that as well.
So ultimately, it needs to be tested against other conditions.
Absolutely.
And really, regrettably, we are looking at still quite a lot of work to be done to confirm it and hopefully improve upon it in various ways.
I think so. I think we've been making progress. I think there's been a lot since I've been involved in this field. We've definitely seen some big movements forwards. You know, there's more people doing research in the area. You know, a test is important, but there might be something we can do if we can work out what's going on in the different subgroups. You know, some sort of treatments would be more in order. You know, a test is good, but if we can understand the biology better, there might be something that we can look at. Now, like ketones, quite interested in ketone supplements. Ketones would maybe overcome that glucose problem if there is a glucose problem. So something that we plan to look at with diet and maybe supplements. But then the disease is so heterogeneous, you need to work out who to give those to, and that is a challenge. That's the big challenge.
Yeah. And then another question is about whether or not the test would be potentially useful even if we don't know the underlying disease process. Well, presumably, yes, it could be, as long as it was proven to be separate to what goes on with other diseases. You know, we don't necessarily need to know what's going on but we do need to be able to show that it doesn't happen in any other condition or even in healthy controls. So it needs to be unique.
Mm-hmm. Yeah, it could be a readout. You know, we don't have very good endpoint measures in MECFS. We use fatigue scores, and it's not very satisfactory. So if the factor was important, and it, you know, say we had a treatment and we tested somebody and we found out they had a big change in their energetics, and then we did the treatment and things improved, and that gives you more and more evidence that something is working, and you've got to sort of read out biologically. So I think from that point of view, in research, it could be very valuable. Also, it gives you clues as to around what might be going on. So yes, I think it could be useful even if it's not a test, and it's giving us clues as to where things are different in the patients. Whether that's the driver of the ME or something that happens downstream is still a big question, and we need to try and tease that apart.
Right here. So I think really what we've covered all that I think is useful at the moment for you know, the subject of specifically something in the blood and your presentation from New Zealand. So something that occurred, which many of us patients were sad to see, was that you applied to the MRC for funding quite certainly, especially in the UK, a large amount for ambitious projects, and unfortunately they turned it down. Probably still a sore subject for yourself, but if perhaps you could, I don't know, give us your view on that, you know, just your broadcastable reaction to it.
Yeah, I was on holiday at the time, which is not good to get that news. We're on holiday. I don't mind giving talks on holiday. My wife laughs because we go to holidays. Where are you giving a talk this time? Yeah, it was disappointing. It's a lot of effort to put together something like that from scratch. We had some really good collaborators. We were going to look at a lot of metabolomics from different cohorts from collaborators in the world. That wouldn't have cost a lot of money relatively to do. And then the key thing really for me about that proposal was to get us going clinically, you know, recruiting patients in Oxford, which I think is really important if we're going to make progress in this illness. So that was disappointing that it wasn't funded. We had various comments back. So 5 or 6 reviewers, 3 were really good, 2 were terrible, and 1 was in the middle. And the 2 that were terrible, we could have probably come back to. They really didn't understand what we were doing. And I think that is a problem with getting people to review these grants. There aren't very many people that see it as a problem. And you're not going to get them to review it. So when you go in an open call, so we had to go in an open call. We went to the populations biology call. We're in with everybody else. It's tough to get grants for anybody, and we're also pretty ambitious in what we were trying to. We wanted a good chunk of money, £2 million, to get going, but we would have gone in what's called a project call. So project grants are normally about 800,000 max, and we were trying to get more than that. And the way the MRC works is that you would normally have a project grant for 3 or 4 years, and then you would build up to a program grant. So we were trying to shoehorn a program grant into a project grant box. And so for them, it was too risky. They had other projects that they could fund. They didn't allow us to come back to the reviewers, which was disappointing because I think we could have made some comments. But I don't think the review process was the problem. I think it was, we didn't have a track record in running, and I don't have a track record running big projects. And the problem I've got is we don't have a clinical researcher that's interested in this space to the level to do research. There's lots of interesting clinicians, but they're not there to do the research. And so that was disappointing. The metabolomics, in hindsight, looking at our new data, not knowing these unknowns would have made it challenging to interpret the data to any pathways. But we would have had an idea as to whether things were different or worth following up on. I was particularly disappointed that we didn't get the money to look at the NIH samples, because I know you talked about the glutamate. They're quite keen for us to help them with their metabolomics. So looking at glutamate in CSF fluid, which would be really useful. But I still hope we can work with them. So we do plan to try and get a paper out, because we don't have much in this space. We have the academic paper, but not any real hard science. Try and get the Polish cohort published with the GET study, and then apply maybe to the Wellcome next summer for a metabolomics grant, including the NIH cohort, the cryotherapy cohort, which you think is a great cohort to work on, to try and get that going. But with the proper, we need a postdoc, we need people to do the science. So disappointing it wasn't funded, but it hasn't killed us off. We're still living and breathing, and we're still carrying on. But it's made us think, actually, it's made us think about how can we do this? So we're not the only ones in this position in Europe or in the world. I had a chat with the Harvard group last week, Ron Tompkins, and we're all struggling with the clinical base. So in the UK, in Oxford, we have a fatigue service. They have patients that come through, so we can access patients. So our plan going forwards is to try and get a 4-center study going with other groups in Europe. So us, Newcastle, group in Spain, Eliza's group, and a guy called Paul in Poland. And we think between the 4 groups, we can recruit probably 50 patients of different classes. And we can do it quickly, because if I try to recruit 250 patients, it'll take me 5 years, and then the worry be about what about the samples in the freezer, are they changing? So we think we can do this over 12 months, get a good cohort together, and the plan is to get a good omics platform. So we would do microbiome, we would do metabolomics, we'd look at L-form bacteria in the blood, and I'll say a bit more about that in a minute, with a collaborator called Soft Cell Biologicals in the US. Maybe Metabolon, if we can get them interested in working with us. And to do this over the 4 centers, we're in discussions with Soft Cell about basically paying for the recruitment at the moment. So this will be a £500,000 project across 4 centers. And you get samples for the first year, and then we would have the samples in our respective freezers, and then we would apply for money to work on them. So without samples, you can't get a grant. So this would allow us to build up that sample set, which I think could be really useful going. And we need to do it, because none of us individually can get to the critical mass to get this to go. But between the 4 groups, I think we can do something quite good. Also, then you've got the interests of the different groups. So we're interested in the factor, so we can pull the samples. The Newcastle group are interested around muscle models, AMPK, activation, and probably PBMCs. The Spanish group are interested in the transcriptomics. And then the Polish group do a lot of good physiology type data. So I think between the 4 of us, we can do something on a world level, which will be for not a lot of money. So at the moment, we're trying to work out what can we do, not cheap, but what can we do with the budgets we've got to work with.
Okay. So would I be right in thinking that with that proposed project model, let's call it, would the reason be that you're looking to source the samples yourselves is so that you're able to have that bit more control over kind of time taken to process, and you're able to ensure that it's the same across all the centres. So you've got, although the samples are collected at 4 different locations, as much as is possible, they've been done in the same way, they've been done on the same time scale, so they can be, as much as possible, directly comparable.
Absolutely. So the timing is important. So for the work we want to do around the plasma, 2 hours we think is max. I wouldn't want a sample that's taken longer than 2 hours to process. So trying to get 50 severe patients in Oxford, we're going to be stretching it, going to London, and we're not going to get that time frame. So there is an issue of doing in different centres, but we need to be careful with the diagnosis. We need to be careful with the clinical measures that we use. But it'd be an interesting test across 4 centres to see if we get the same data. If we collect samples in Poland, even if we only get 10 severes, are they the same as our 10 severes? So we can do that interesting correlation. So soft cell biologicals are finding some interesting things in the blood in chronic illnesses. So this is across the board. So ME, fibromyalgia, endometriosis, migraine. There's high levels of what are called L-form bacteria in the blood of these people. And so they've done this in America, and they're finding in different parts of America, different controls have different species. So, that would be very interesting to see whether is there a bug in the blood that correlates with ME/CFS, or is it just variable? Is it just the fact that you've got bugs in the blood that makes a difference? So they're quite interesting in this sort of multi-center approach for what they're doing, but also the microbiome. Is there a difference in Spanish one and a UK one? And we can get that data, but I think using the right diagnostic criteria, there's this Ficada or the new, do you go for it? But you need a PEM element, and see most of our Ficada patients from Poland have PEM. About 95% of them have PEM. So, but knowing the ones that don't is quite useful when you know that they don't have it going into the mix. So, I think, I think it could be, we can get around the barriers, but setting up everybody working on the same sheet, recruiting the patients the same way would be great.
Can you expand a little bit more on the difference between L-form bacteria and which is probably a term that most, if not all, viewers probably haven't heard of before, and normal bacteria?
So bacteria can have walls. So the ones that are sensitive to penicillin, you go to the doctor, they'll identify those. And there's gram-negative and gram-positive bacteria. So a bacteria with a wall can exist in the body in an L form. So an L form hasn't got a bacterial cell wall, it's just a membrane. In a way, it's like a mitochondria, which is quite an interesting way of thinking of it. So it's in the blood. So things like mycoplasma, of course, sort of TB, that type of thing, they don't have cell walls, so that would be like an L form type bacteria. But what Brent's finding is that many bacteria that don't have known L forms can exist as an L form in the blood. So they may be in your gut as a wall forming bacteria, but in the blood they're an L form. And those L forms aren't normally very antigenic, so they don't trigger an immune response as much as one with a wall. So in these diseases, you don't see high levels of inflammation, you don't get markers of inflammation, but they have these bacteria there, and they're in normal people as well. So there's a difference. There seems to be a difference in the number of strains found in people with chronic illnesses. So there are specific antibiotics that work on L forms. So you wouldn't treat an L form infection with, say, penicillin, because it wouldn't work. But there are other antibiotics like doxycycline, which again is a drug that impacts on mitochondria, which is another one of my minocycline. These all work well on L form bacteria. So I know that the guys in Utah are thinking of running some studies, finding out what the bugs are in these patients and then culturing the bugs. So what they've managed to do, which is no one ever's done before, is to take these L forms and culture them. If you say to somebody you've got bacteria in your blood, people would laugh. And I think people don't take them that seriously, but they're able to culture bacteria in the blood. And then they can test antibiotics on those cultures and then try them in the patient. So you can actually see whether you have an impact. So it's an exciting area of science, and it's quite new. You can look at the website. Not much is on there, but it just gives you a flavour for what they're doing.
Okay, brilliant. Thank you. So you've described this multi-center potential project. Do you have, and you've also mentioned at least one other project already earlier on. Do you have any more that you can talk about, or?
Yeah, so for me, trying to recruit patients in Oxford is the key for us. If we can start doing that, it's not going to cost a huge amount of money. We've got the support of the MET clinic, and they'll help us. So if we get this 4-center study going, so we have some ideas. So Jamie Strong in my group has been looking at a pupil monitoring device. The group in Poland are looking at aortic stiffness. So the vasculature may be different in patients. The smooth muscle that makes up the sort of iris of the eye is an autonomic reflex. And he looks at himself, and his response to light is really quite slow. So we have just taken on a placement student to develop this. So we need to write proteins and programming. So that's a great project. It wouldn't cost a huge amount of money to develop this device with the idea of using it clinically. There's somebody in Oxford that's got a big grant in the stroke area, Alzheimer's area, to do something very similar. So there are devices out there that you can measure the pupil response, sort of clinical GP type. But this would go on a phone. This would be on a phone app. So the idea, you take it home, you could maybe monitor what your iris is doing and work out whether you're going to go into PEM or not. We don't know, but that's an area. And the other one is with a dove clinic. I've done a bit of work with Julian Kenyon, who I really rate very highly. He's a smart guy, and he's done some fecal transplant work. And we'd like to do a proper clinical study with him with placebos, pre-select the patients by looking at their bacterial load in the bowel, do the microbiome, and also L-form load, and also do the metabolomics to see, and to really pick out patients that we think could really benefit from that. He's doing some stuff at the moment, looking at that in a few patients, but so that's something. So that will be a bigger project, but we've got someone that can recruit patients, so he's able to do that, and that's what we need in Oxford as well to do the other bits and pieces. So.
And in terms of funding for those, will you be needing to apply to MRC or other funding bodies for those, or are you exploring?
Yeah, there's a few options. So we're thinking of maybe looking at crowdfunding, but I think we need to get a European grant going. I want to get that going first. So, because I need to keep my people. So my scientists need to be in the lab and have a reasonable duration of contracts. I've got one at the moment on a 12-month contract, and getting more people to do that is going to be challenging. So getting the 4-center study going first, and then I think we could put some projects up for crowdfunding. It's a little bit tricky with the ethics. So normally what you do when you apply for ethics, you need a grant. So we would need a grant in place, and then we'd apply for ethics. But obviously with crowdfunding, we don't have the grant. So I'm a little bit uncomfortable asking somebody to support a project we'd like to do, say for the fecal transplant one, and then we don't get ethics. It's unlikely to happen. Then how do I give the money back? So it's that sort of worry. For the one with the pupillometry device, they're not going to not give us ethics. And actually, I've made inquiries about the fecal transplant one, and we should get ethics to do that. So that's my main concern. So crowdfunding is one route. You may get donations coming in to fund these projects. But the idea is we have a website that we've been developing, is to get that to go live. We've been so busy trying to keep going, publish papers, it's not gone live. So that will put the projects on the website, and people can do that. And I've talked to the university, they will let us do this. So it's always tricky to get them to do things like this. But I think they're quite supportive around the ME space. I think they see a need as well. So.
Okay, so really it's the message for people out there interested in supporting your work is keep an eye on the website once it goes live. Mm-hmm. And naturally on the forum we will feature that when it does happen, and obviously we'll notify people via our social media channels as well. So but once that website is up and running, then it's to keep an eye on that, and if if they're keen to support your ongoing work, then that would be one potential way to do that for them.
Yeah, absolutely. Yeah. And we can earmark money. We can put it in pots for specifically for ME/CFS. Begin to get the patients. So I think the patients perhaps would like to fund specific projects rather than a blanket. We're going to work in this space. I think we can do that. We've got some interesting ideas. We're not short of ideas of what to do, and so we can have different levels of project. The pupil one could be done for about £20,000. The fecal transplant, we're looking for more 100, and we can get for match funding. We get some money coming in from patients. There are various pots, particularly within the university. If I've got half of the budget already, I'm more likely to get that money. So for these smaller projects. MRC, we might go back. I think we need to publish some papers.
Sure.
And if we can get a few things out, I think the Wellcome Trusts, we've had a chat with them, and I think they'd be interested in a project, not a big, huge £2 million project, but something to build on the metabolomics.
Okay, brilliant. Well, I'm right out of questions.
There were quite a few questions, actually.
Yeah, I'm afraid so. Sorry about that. So yeah, well, we'll wrap it up. If there's anything else that that you'd like to mention at the moment, then please feel free. But
no, I think we're keen to carry on in this area. We put a bit of effort into it, and and it's going to be slow. I know that it's going to be slow to move forwards, and there's lots of other areas that we're interested in that could have overlap. So we've done some work on brain death models in rodents, where the brain dies, causes catastrophic problems with kidneys that are going to be transplanted, and the energetic problem is there exactly the same. It's very similar. So I think looking outside the box, looking beyond ME/CFS, and it may be that we apply for money in those areas and then bring in the ME plasma samples as a comparator. Same in stroke. And I know Newcastle are thinking the same way. They do a lot of work on Sjogren's syndrome, and so if we can't get money for ME/CFS, we have to think a bit lateral and maybe think, well, we can get money for this. Let's try and use. So for our MS is our fatigue that's not ME/CFS, and maybe we do things the other way around. So we're keen to keep going, and I think we will. And it's good to get. I do have a look at the forum. I'm not keen on joining forums, but I do get a lot of useful stuff. I follow. I like is it John Edwards' comments? I think they're on the ball. I quite like reading some of those. So it's and also you get the papers before I find them, so I really quite
Well, it's good to hear that we're of use, as in the forum is of use for more than just the patients. It's one of the things that we've attempted to do, which is I suppose you could describe as trying to provide a bridge, some kind of link between people who do. A lot of people, patients,
sure,
find it harder to interact on, say, more formal ways. So it's nice to know that.
One of the things we plan to do, whether we could work out how to do this. So when we publish the paper around the Polish cohort and the GET study, we could maybe do a presentation on that on an open platform. There must be ways of doing that the patients could watch, and even if they write questions down, we could have a follow up and just address those. So it's quite fun to do. But it's good to get the impact. And you were talking about your ideas around trying to work out subgrouping the patients, trying to put them into different groups. And I think for our clinical studies, that could be really useful, taking what you've found by being patients and taking that on board, rather than just asking somebody, are you tired? I think there's other ways that could be useful, particularly if the clinical differences in clusters is informative. Then there may be some indicators from those clinical variables that you will see as symptoms, which we could tap in on. So
great, brilliant. Well, thank you very much for your time. And we'll look forward to seeing that website going live. Again, like I said, we'll make sure as many people know about it as possible, and look forward to speaking to you in the future.
Okay, thanks, Andy.
All right, thank you.
Okay, bye.